Hello everyone, I'm Seongjin Choi, member of IMEC, and today I will present the preprint titled Topology identifies concurrent cyclic process in single-cell transcriptomics and androgen receptor function. Here's the abstract, so I'll give you a minute, so please read this abstract. Okay, so could you find the gap sentence?
The first one?
Sentence. Yeah, also I agree. First sentence is the gap sentence. So let's dive into it. So single cell RNA sequence data tell us which genes cell in a tissue is turned on, how much. For example, this is your tissue, and we have a pre-process such that cells are separated, and we put in the gene so that each cell is enveloped by a gem bed. So each gem bed has exactly one cell, and this cell has some mRNA. So the remarkable thing is that each gem bed distinguished which mRNAs come from. Based on this, so we can have some cell gene matrix. This means that the number of mRNAs from each cell. So before that, we must pre-process the single cell count matrix, but I want to talk about that. So after pre-processing, the number of mRNAs for each cell is counted. So this also could be regarded as a point cloud in RM, but in practice, we use M is too large to deal with, so we use PCA to reduce the dimension. But problem is that single RNA sequence data is a snapshot, single moment, so making hard to detect periodic activity. For example, here is the figure for cell cycle. So this is clearly form a cycle structure. Also, we assume that tissue contains sufficiently many cells so that cells are well distributed along the cell cycle. So in this case, when X is the cell gene matrix, then we consider PCA matrix from the raw pre-processed single-cell RNA sequence data and remaining top three PCA value. Then we can also consider it as a point cloud in R3. So consider the collection of genes relevant to G1 cell cycle procedure. Suppose that we have such a circle value function on that kind of point cloud, then this would be a great candidate for that indicates the phase of the cell cycle. So the problem is that how to drive such a circle valued function from point cloud single cell RNA sequence data is the main motivation. So such a circle valued function is expected to be continuous. So I will explain it. Assume that such circle valued function exists, then it reflects the biological phenomena. Then a slight change of the domain is also a slight change of its value because it reflects the biological phenomena. That means that circle valued function should be continuous in some sense.
We have some question in the chat. 행이 준이 아니라 세포여야 되는 것 아닌가?
그 이전 슬라이드에서 여기서 이 PCA 할 때 그 이제 PC를 이제 그 진 차원을 이렇게 줄이는 거잖아요. 그러니까 이제 PC가 결국에는 그 진의 피처를 이렇게 줄이는 건데 그러면 또 이게 행이 셀이어야 되지 않나는 질문인 것 같아요.
At this point represent cell and each vector component represent PCA component.
So the row each row means the one cell, one cell, two cells in a protein.
Yes. Sorry, it's error. Sorry. Yeah, it should be cell. Yeah. Yeah, it's error. Yeah. So PCA1, PCA2, PCA3, and cell 1, cell 2. Yeah, this should be correct. So topology on the point cloud is required when we require that circle valued function is expected. But the main problem is that the topology of a point cloud is disconnected and too simple. Let me give some famous example. So consider the point cloud on circle. So we expect that we argue that this is a circle because it's based on circle. So the problem is that when we compare two space, so the most convenient way is to compute some algebraic invariant called homology. So in this case, I will explain later. But yeah, the 0 degree is just the number of points. So with dimension, number of points. So, but higher dimension homology vanishes for k greater than 0. But on the other hand, in circle case, homology is quite different. So it survives for degree 1 homology or cohomology case. So this means that even if we allow to put into many points on the point cloud, this difference cannot be resolved. Yeah, this is not regarded as same. So we must upgrade the point cloud containing high dimensional homology. So the mathematicians invented a way how to tackle it. So this is called the Rips complex. So let me give some examples. So left figure is the represent the point cloud with where epsilon is indicates the epsilon ball. So we thicken the point cloud with center at each point in the point cloud. Then when it varies the radius for epsilon, when epsilon varies, then the topology changes. And for example, when epsilon equals 5, then one-dimensional objects appeared. When epsilon equals 7, then two-dimensional object or three-dimensional objects appeared. So this means that there is a possibility that two-dimensional or three-dimensional homology survives. So, but epsilon equals 0 or infinity case is not very interesting. So also, Rips complex is defined as follows when we collect all simplex when each pair of the point is less than epsilon. So from this condition, we can easily see that this condition is satisfied. This means that this Rips complex is a Simplicial complex. Why Simplicial complex is nice? So I will, in the next slide, I will introduce how to compute some homology on simple complex. So it's a nice algebraic invariant containing information about topological space. So idea is that we will use the first homology class of the Rips complex to get a circle valued function we described earlier. So that's the main idea, but it's quite abstract, but so sorry about it. Yeah. Oh, sorry. But simple complexes are topological space which homology is to compute. For example, left figure is the example for simple complex. So, for example, X0 is the collection of zero simplexes for its dot. So V0, V1, V2, V3, V4 is the collection of zero simplices. Also, we give a total order on the zero simplices. Then one simplices is the collection of one simplices as follows. And there is only two simplices colored by yellow, and there is no high dimensional simplices. So this simple complex is just consists of 0, 1, and 2 simplices. So in this case, we can consider chain complex. Let me give some example. For zero cochain contains V0 twice. So this kind of formal sum are all elements of the zero cochain, zero chains, space of zero chains. For example, V4 minus 12 V2 could be also an element of the C0. For example, C1 could be formal sum of V2 V3, V1 V3, or V3 V4. What is the boundary means that this V1, V2, V3 indicates the yellow two simplex. So boundary is defined as follows. So it considers the orientation of the two simplex. So this sign represents the orientation. So V2, V3 has a plus sign, so this direction. But V1, V3 is a minus sign. So this is the boundary. But this is quite similar, but V1, V2 minus V0, V2 plus V1, V0, V1 is a one cycle in this case, but it does not come from the two simplest. So it's not a one boundary. So you can see that boundary must be cycle, but conversely is not true. So homology measures how many n cycles not boundaries in the simple complex. So in this case, I only consider the degree 1, but we can do for any degree. So that's the meaning of n degree n homology.
Oh, sorry. How can we connect the notion the Rips complex? The Rips complex in previous slide they defined with the matrix.
Yeah.
The between distance and here we have kind of direct.
It's because Rips complex is simple complex. So when we have a tool to compute the homology of simple complex, we can also compute the homology of Rips complex. So that's that's what's going on.
Sorry, but I'm not quite friendly with simple complex.
For example, yellow when we have a two simplex, then its subset should be also simplex as well. For example, V1, V2 should be also simplex. Also, since V1, V2 is a simplex, V1 should be also simplex. That's the definition of simple complex. If some simplex is contained in X, then any subset should be also simplex as well.
I focus on algebraic continuous compressive bias on silly question. Maybe this setting the order of vertices is essential to compute boundary.
It's quite essential.
Is this affects is the choice of order will affect homology.
I don't think so. It is irrelevant. Yeah.
So when we using TDA, then we can arbitrarily choosing the order of point.
Yeah. Yes, it's the explanation of simple homology. Also, what the intuition is that we decompose the space into several smaller pieces. So what we get from that information, that's exactly what homology says. But however, there is another way to study space by considering its defining function on the space. That's the idea of cohomology. Simple cohomology contains information about functions on the simple complex. So why do we also consider the cohomology? Because we are interested to find circular valued function based on simple complex. So that's why we need cohomology. So same setting, then slightly changes that we consider not space itself, but function defined only. So for example, so C0 is the degree zero space of degree zero cochain is the function from the vertex to the C. C could be changed to any other ring. So C1 is the function from the one simplest collection of one simplest to the C or any other ring. Also, in this case, we can also define some boundary operator. So same situation happens. So degree n cohomology quantifies how many n cocycles, but not n boundaries in X. Some non-trivial theorem is that when the ring becomes filled, homology and homology are isomorphic as vector space.
So F should be homologous. Is there some other?
Yeah. In this case, yeah. So persistent barcode records how the first homology class changes as case varies. For example, so cycles will be generated when we varies the scales, but it could be that also when we enlarge the scales. So that's the barcode exactly captures that moment. In fact, it's hard to compute by hands, but there is a program to compute such persistent barcode when the point cloud is given. Also, in this case, we should be work on the finite or not should be finite field for such barcode because of some theory. In this case, this barcode means that B1 is the first Betti number. This y-axis means that the first generators for first homology. So this bar is the longest one in the first homology. This means that some first dimension cocycle persists long. So we choose some cocycle from that barcode. Also lift this cohomology to C integer value coefficient. So yeah, some people said that in practice it's always possible even if it's impossible, we can vary prime number so that we can almost always lift to the coefficient. Then we should find some harmonic representative of that cocycle. This is complicated, so I will not talk about it, but we can do it also by computer algorithm. So we can find harmonic representative. So after that, using that harmonic representative of the first cohomology class, we can define the circular coordinate. So it's hard to write down by hand. Anyway, it's possible. Also, some theorem guarantees that it is continuous on simple complex. So when you restrict to the zero simplices, then it's the desired circular valued function.
Oh, can I have a question? Oh, okay. So, I suppose like the lines corresponding to B sub 0 represent the black dots we initially have.
Yeah.
And then they kind of like died when dots are connected by the line segments when we increase the radius of yellow circles.
Yeah.
And it seems like B sub 1 contains different barcodes correspond to triangles, which are too simplex.
Yeah.
Right? But why don't we have barcodes corresponding to one simplexes, like a line segment?
Because it is not a cycle. Line is not a cycle. So as I explained before, so some closed formal sum of the closed form would be a cycle. So line is not a cycle. So this would be not recorded as in a barcode.
Then what about the tetrahedron? Is the tetrahedron a cycle, like two-dimensional cycle?
Yeah, right. That's right. Yeah, but in this figure, we only recorded so one-dimensional case.
Okay.
So one dimension. So yeah, you're right.
P the field.
Yeah.
So is it kind of image of the functions to define over
Yeah.
for me,
Some to consider the such persistent barcodes, so we must work on the field. So, but we are for some reason for lifting, so we will consider the field finite field related to the prime number. So P is prime number.
Also, is it
Yeah.
necessary for persistent homology?
Yeah, both of them. Yeah, they are isomorphic when this is filled.
Why you get that field to define work?
It's because in fact we have a filtration and there's some theorem called theorem about persistent module. So it could be decomposed into several persistent intervals, but it only holds for when base ring is filled. Yeah, it does not hold for ring of integers in general. Yeah, so far, anyway, we found the circular coordinate on the point cloud. So based on this, we can also compute the phase or amplitude for each gene when you assume a chain of offset models. What is chain of offset models? So let me consider the following diagram. So sorry for error path again. So it should be cell, and we have a circular coordinate from by complex procedures. So I will call H as some circular coordinate. So with this circular coordinate, we might find some loop in that space we are interested in. Also, when it's cell and cell, then consider the just embedding to that gene space. So cell 1 goes to cell 1, not PCA vector, but original gene vectors. So this is the map of this embedding. Based on this, suppose we have a loop in that, and this embedding, we get a loop in the gene space. So when chain of offset model assumes that this loop should be a form like this. So this chi is the periodic function. So it will be sine or cosine. So we are interested in. So because we are only interested in first harmonic approximation of the periodic function. Also, BEI would be the base for the periodic phenomena, and AI would be some amplitude of the biological phenomena. So what we want to do is to find AI and BI in the chain of offset model. So how to find such model variants? So this is achieved by the compute-lead-lag matrix. It is some computable. So when gamma is given, so it's just an integration. So it is in the appendix, there is a theorem that this A and B can be written as some not closed form, but a closed form of eigenvectors of the lead-lag matrix. So by solving linear algebra problem, we can get some A and B. But problem is that we need such L to define the lead-lag matrix. So the other assumes that some simplicial lead-lag matrix, they used simplicial lead-lag matrix rather than original lead-lag matrix because it does not require any choice of L. So it only depends on the choice of circular coordinate and this embedding. So others also insist that they have similar roles, so it does not have a problem in the discussion section. So
here next for every gene.
Oh yeah. It's in fact it's an error of cell 1, cell 2, cell M, and cell 1, cell 2, cell M.
So here we give I for every gamma here.
I for
so gamma to the I here.
Aha.
Yeah.
Yeah.
Every gene or cell?
Each gene.
Each gene.
Yeah, each gene.
Okay, so we make a circle.
Yeah.
For every cell.
Yeah, that's the strong assumption.
And then every gene also have its own cycle.
Yeah, right. Exactly.
And according to that's whole space cycle, some genes can be lead or lag.
Yeah, right.
Phases. So it can the lead lag matrix compute that relationship and
exactly. Right. That's right.
Then that the mapping H does not exist. Cell has no cycle.
Yeah, but when we choose, but when we vary the epsilon radius, then in practice there must be a cycle, one cycle. Yeah. Yes. So there are lots of points in the point cloud. Yeah.
I have one more.
Yeah.
So we give circular coordinate for every cell here. So the barcode keep changes that when we change the F. We fix one epsilon to technique.
Yeah. In fact, we choose the longest barcode.
I see.
Yeah, and in that range, it does not matter.
Right.
Yeah. Okay. So far we get circular coordinate firstly we must fix a collection of set of genes we are interested in. So we expect that we expect the circular biological phenomena in that kind of cells. So we fix set of genes and project or restrict to that vectors accordingly, and we get a circular coordinate for it based on this PCA top three components. And also we get a by assuming chain of offset model, we also get some periodic model for genes simultaneously. That's the situation. So others applied it to several data sets. So claim is that estimated gene bases recover the known S to G2M ordering of the cell cycle. So here is the so AI was the amplitude for the genes, and BI was the phase. So this is the angle for phase for the some circular behavior, and it matches the biological phenomena. So they use the human dermal fibroblast single cell RNA sequence data set, and what we are interested in is the set of genes related to S and G2M bases. Also, H is the circular coordinate, and XIC is the number of pre-processed number of genes related to I. So that's the graph for S genes and G2M. So in this case, you can see that the peak comes earlier in S genes compared to the G2M case. So it supports that their framework works well. Also, so they analysis studied furthermore because AR is known to repress some cell cycles. So they consider the following. So it is the previous single cell RNA data cell, but they made another new single cell RNA data set by injecting some shRNA to make AR silence. Also for control group, they also injected shRNA but scrambled one. So for 3 cases, we get circular coordinates for cells and amplitude and phase for genes for each case. So compare the results with the original HDF in the left case. So here is the result. So R value for the original HDF case and some HDF with injected shRNA sequence silencing case, but their cell cycle, so amplitude and phase are very overlapped in this case because R values are very high. Even more, so they compared some HDF with shRNA silencing and with control. They are very highly coincide. So this means that AI status does not matter for cell cycle in this case. So they conclude with this analysis. Also, so lastly, they studied the data set from the mice. So mice were castrated and given the HDF at 28 days to induce prostate regeneration with tissue sampled over the following days. So in this case, also we get a single RNA sequence data, and we have 50 collection of some interesting gene sets. And for each gene set, we have a persistent diagram we discussed. Based on this persistent diagram, we can compute its ring score. This is defined using persistent diagram, and it indicates that larger values indicate more dominant circular structure, because it says that some persistent first degree persistent barcodes are large. So based on that, they invented some p-value like value. So for example, is that ring score is important to consider as to find cyclic procedure. So they randomly select n genes and compute the also ring score and repeated randomly generated gene sets with fixed n and create new distribution. And with this new distribution, estimate the p-value for the target set of genes. That's how they got p-value from the right column. So based on that, they concluded that androgen response, cholesterol homeostasis, and myogenesis are highly plausible to get circular phenomena. Also, they are interested in whether they are dependent or completely dependent or independent. So they concluded that if they are totally independent, then this point should be equally distributed. Or they are completely dependent, then it should be not the torus, but it should be circle. But neither is the case. So they concluded that androgen response and cell cycle are partially related. Same holds for cholesterol homeostasis as well. So here's the summary. So when we have a single cell RNA sequence data with a suspected set of genes that would contain some circular phenomena, then computing persistent cohomology would give some circular coordinate on that point cloud. And with another assumption of chain of offset model, then this could be used to reveal a cyclic pattern in single cell RNA sequence data. Yeah, thank you.
Well, I have